Welcome to part 2 of our lesson on drug discovery and clinical trials. In this lesson, we will be looking at the different phases of clinical trials from phase 1 to phase 4, and we will also touch on the role of IRB in clinical trials. The drug discovery process generates multiple hits, which are compounds that show the desired biological activity. After further testing and optimization, the most promising compound, which is known as the lead compound, is selected to proceed to preclinical research in animals and clinical trials in humans, with the aim of entering the market as a drug. First, let's take a look at preclinical testing. Preclinical testing is carried out in the lab, where the drug is tested on cells and animals. Preclinical testing checks the pharmacological activity of the drug to find out its effect on the body. The ADME characteristics are evaluated. ADME stands for absorption, distribution, metabolism, and excretion of the drug. At the preclinical stage, the tests are carried out in lab-grown cells or in animals. Tests that are done using cells are called in vitro studies. In vitro is Latin for in glass. Tests that are done using animals are called in vivo studies, where in vivo is Latin for in living. In preclinical studies, the toxicity of the drug molecule is studied extensively to determine whether it is safe to test in humans and to help determine a safe dose to be tested. During preclinical testing, the manufacturer will also need to do other concurrent studies, such as chemical studies to establish the drug's purity, stability, and shelf life. The manufacturer also needs to plan how to scale up production to prepare for clinical trials and eventual distribution if the drug is approved. Dosage form design. For example, will this drug be formulated as a tablet or injection or inhaler, etc. If the results from the preclinical testing are promising, the company can then apply to proceed to the next stage, which is testing in humans or clinical trials. At this stage, they will file an investigational new drug application or IND, and the lead compound will change legal status to become an IND. Now let's take a look at clinical trials. The objective of clinical trials is to determine whether the drug is safe and effective for the patients. Before a drug is approved, it has to first go through phase 1, 2, and 3 clinical trials. After approval, phase 4 studies may still be required. The different phases of clinical trials differ in terms of their number and types of participants, the duration, and the outcomes of the study. Let's take a look at the objectives of each phase of clinical trials. Phase 1 trials are conducted to assess the safety of the drug, and it is tested in healthy volunteers, not sick patients. Phase 2 trials are conducted with patients with the particular disease. In addition to safety, the efficacy of the drug is also evaluated. If phase 2 results are promising, the drug is tested in a larger group of patients in a phase 3 trial to confirm the efficacy and safety of the drug. Based on the results, the manufacturer can then apply for approval to market the drug. Phase 4 trials are carried out after the drug enters the market, where the efficacy and side effects of the drug are monitored in the real world. Now let's take a look at each phase individually. Phase 1 trials involve about 20 to 80 healthy volunteers, and the volunteers are not blinded, meaning they are aware that they are taking the drug. The purpose of phase 1 trials is to assess the safety profiles, drug tolerance, ADME, dosage, and side effects of the drug. Typically, about 70% of drugs that enter phase 1 trials make it to the next stage. Phase 2 trials involve about 100 to 300 patients with the target disease. These studies are typically double-blinded, randomized trials with placebo control. Some studies may employ a crossover design. I will explain what these terms double-blinded, randomized, placebo, crossover mean in a few slides ahead. Phase 2 trials determine how large the therapeutic effect is, as well as what is the optimal dose and dosage form, and what are the side effects of the drug. Since phase 2 studies are quite short, typically lasting a few months, only the short-term side effects will be detected. At this point, long-term side effects are still unknown. Typically, about 40% of drugs that enter phase 2 clinical trials will make it to the next stage. Phase 3 trials are conducted with several thousand patients in multiple centers worldwide. Similar to phase 2 clinical trials, most phase 3 studies are randomized and blinded. Since phase 3 trials are carried out on a larger population and over a longer duration, it can provide data on long-term safety and identify uncommon adverse drug reactions. Approximately 70% of drugs that enter phase 3 trials will successfully enter the market. After the drug enters the market, phase 4 trials continue to monitor the efficacy and side effects of the drug in the real-world environment. So here's a summary. Phase 1 clinical trials are carried out in a small group of young, healthy people. It tests for possible harm and side effects and determines a safe dosage of the drug for further testing. Phase 2 clinical trials are carried out in a few hundred patients with the disease to determine whether the treatment is effective and has side effects compared to a placebo. Phase 3 clinical trials are carried out in up to thousands of patients over long periods and over many different countries. It may be compared to placebo or against other existing treatments. If the drug is deemed to be safe and effective, it is then licensed to be sold. Phase 4 trials are carried out after a drug is approved to monitor the effects of the drug in the real world. In most phase 2 and phase 3 trials, some patients will be allocated to take the drug and some will be allocated to take a placebo. A placebo is made to look like a real drug, but it is actually made of inactive substance. Why do we need a placebo? Because the mind is very powerful. A patient who believes that they are taking a powerful drug can actually feel the improvements in their body. So it's important to ensure that any effect is really due to the drug and not a placebo effect. In a trial, there are patients and there are investigators who will examine them. So who knows whether the patient is taking a drug or a placebo? If only the patient is unaware, but the investigator who examines them is aware of what the patient is taking, then it is called a single blind design. Because only the patient, a single person, is blinded to the treatment. But ideally, the trial should be double blind, where both the patient and the investigator do not know what the patient is assigned to take. This ensures that the investigator is not biased when collecting the data. Now let's take a look at some of the terminology used in clinical trials. First, what is a trial group versus control group? In the clinical trial, the trial group are the people who receive the investigational new drug, whereas the control group may either receive a standard existing treatment or a placebo instead. Now let's look at blinded. As we saw earlier, single blind means that the patients don't know which treatment group they are in, but the investigators know which group they are in. Double blind means that both the participants and the investigators don't know who is in the trial group or who is getting the IND. Blinding is very important because it helps to cancel the effect of bias in the way that the patient and the investigator responds to the treatment. Next, what is randomized? So randomized means that the patients are allocated randomly into the trial group and the control group, and they do not get to choose which group they are a part of. Some clinical trials have a crossover design, so that means initially some patients would take the placebo and some would take the drug, and somewhere at the midpoint they would switch over. So those who were initially taking the drug would then take the placebo, and those who were initially taking the placebo would switch over to the drug. Let's take a look at an advertisement recruiting volunteers for a clinical trial. Which phase of clinical trial testing do you think this ad is for? A keyword here is healthy volunteers, so this is most likely to be for a phase 1 clinical trial. Now how about this ad? This ad is looking for patients with diabetes, so it could be phase 2 or phase 3 trial. There's no further information regarding the duration of the study or how many participants in total they are recruiting, so it could either be a phase 2 or phase 3 clinical trial. After completing the phase 3 clinical trials for a drug, the manufacturer can then file a new drug application or NDA, where they request for the license to market the drug for the specified indications. After a drug enters the market, the pharmaceutical company will continue to collect data on the safety, efficacy, and optimal use in the population. This falls under phase 4 clinical trials or post-marketing surveillance. The purpose is to report rare adverse drug reactions, product defects, drug interactions, and determine whether any additional warning or information should be added to the label. Depending on the severity and frequency of the side effect, the drug may even be withdrawn from the market. When a new drug enters the market, it is protected by a patent so that the manufacturer has the exclusive rights to manufacture and sell the drug. After discovering a new drug, manufacturers typically apply for a 20-year patent. However, after completing preclinical testing and clinical trials, only a part of this period remains. The drug manufacturers can extend the length of patent protection in several ways, and typically pharmaceutical companies are very good at extending the patent protection by filing multiple patents on the same drug. Patent protection enables the manufacturer to receive a good return on their investment, and therefore it provides an incentive for them to invest in drug development. After the patent expires, other companies can then come in and produce generic versions of the drug and sell them at a lower cost. In this lesson, we have looked at clinical trials. But who ensures that the research is conducted in an ethical manner and safeguards the interests of the subjects? The IRB, or Institutional Review Board, consists of a panel of doctors and other healthcare professionals like pharmacists, nurses, etc. in the institution. The role of the IRB is to ensure that ethical principles are adhered to whenever decisions are made affecting patients and volunteers. Any new drug trial needs to be approved by the IRB before they can be conducted. This helps to safeguard the interests of the human subjects of the trial. We have reached the end of this lesson, and I hope this gives you a clearer idea of the different phases of drug discovery and development. Thank you.